Good morning and welcome to Emerald Ash Borer University. This is Robin Osborne from Michigan State University. My EAB University colleagues are Professors Cliff Sadoff and Elizabeth Barnes from Purdue University and Amy Stone from Ohio State University Extension. And today we are happy to share with you the presentation entitled Responses of Non-Native Species to Climatic Change and Their Implications for Management by Sam Ward, entomologist at Mississippi State University. Sam did his graduate studies in entomology at the University of Minnesota, where he worked on the biological control of emerald ash borer for his master's degree and the resurgence of larch casebearer in Minnesota for his PhD, which you will be hearing a lot about today. He then spent 2 years as a postdoctoral research associate at Purdue University, where he researched landscape scale drivers of invasion by forest pests. In August of this year, he became assistant professor of insect ecology at Mississippi State University in the Department of Biochemistry, Molecular Biology, Entomology, and Plant Pathology, where his lab is beginning to work on the ecology and management of several native and non-native insects. We welcome your comments and questions today, so please feel free to type them into the chat pod or the Q&A pod. Sam will be answering them right after his presentation. Tomorrow, you will be mailed a link to a short, voluntary, confidential survey that we hope you will take the time to fill out. It helps us bring these free educational webinars to you. The email will also include our presenters' contact information, as well as information on how to obtain CEUs for viewing this live webinar. This webinar is being recorded and will be available for viewing later on the EAB University page of the www.emeraldashborer.info website. Thank you for attending today. And Sam, please unmute your mic and you can begin your presentation.
All right, thank you very much for the introduction, Robin, and thank you to Cliff Sadoff and EABU in general for the invitation to talk about some of the things that I work on and think about. And as Robin alluded to, if I can get my screen to advance, there we go. As Robin alluded to, I'm going to be talking a lot about larch casebearer today. That'll be pretty much 90% of where we spend our time. So if you haven't heard of larch casebearer, you're going to get probably more than you ever wanted this morning. And then in the spirit of EABU, I'm going to talk a bit about the invasion of emerald ash borer and kind of some things I've started to think about since I've moved to Mississippi. Before I dive into everything, I just want to acknowledge all the people that have contributed to this research along the way, such as Songlin and Sandy during my postdoc. And then a bulk of the work was done at the University of Minnesota with Brian Alkema, and had a lot of help with field work and all sorts of different things from the people in the lab there. And I also have to give a shout out to Aubrey Keys. As you'll see throughout as I present these things, there's a lot of work done, and definitely none of it would have been possible without a lot of her help. So I definitely want to thank them, and then also thank all my funding sources here on the right. So to dive in, talking about larch casebearer, Coleophora laricella. It's in the family Coleophoridae. So it's a moth when it's in the adult stage, and I'll go into the life history in detail, but just to explain this picture a bit, this is a tiny third instar larva that's feeding on larch foliage. So there's an insect inside that little cigar-shaped case. Before I get into life history, though, I'm going to talk a bit about the invasion history of larch casebearer. So larch casebearer is native to Europe, where it feeds on European larch in the mountainous regions. But it was discovered near Northampton, Massachusetts in the 1880s. Subsequently, it spread throughout much of the range of eastern larch. The range of here is depicted in dark blue. Eventually, it made it over to western larch, detected there in the 1950s, and spread throughout most of the range there. And I'll talk about the invasion in both of these regions, but I'm going to start with some of the patterns that we've seen in Minnesota. So if you were to go into a bog in Minnesota, you might find eastern larch or tamarack. Larix laricina. Yeah, so these trees occur in seasonally flooded sites. They're horrible places to do field work in because if you've ever been to Minnesota and around standing water, there's lots of mosquitoes. But it's really a wonderful time of year or really wonderful places, these bogs, to be in autumn when these trees turn golden brown and lose all their foliage. So while these trees look like a spruce or a fir, they have a more similar life history to an ash or a maple, in that they lose the entirety of their needles and turn a very golden brown, beautiful color, which I'll have some pictures of later on. But that tree life history, something to keep in the back of your mind because it'll become important as I talk about some of the ecology of larch casebearer and some of the things that we noticed in Minnesota. So if you were to go into an infested tamarack stand in Minnesota, you might find these tiny silvery gray moths. So this is the adult stage of larch casebearer, typically abundant from spring through summer. The moths lay their eggs singly onto larch needles. So right here is what looks like an upside down tiny little jello mold. And that's a single egg from larch casebearer. And then this defoliation that you see here is actually from an early instar larva that's hatched from beneath or through the bottom of that egg and has begun feeding inside the foliage. So these are very, very tiny, minute insects. So as the insects develop, as the larva develops, it eventually will cut off that needle that it's been mining in. This happens sometime in late summer, early autumn, and then they bear it as a case, hence the name larch casebearer. So this is a hollowed out larch needle right here. You can maybe see some of the silk strands down here. So that larch needle gets lined with silk. The insect is then in that case for the rest of its life cycle. And so this is the head capsule of the insect down here, walking along a single larch needle. As you move into winter, these larvae attach their cases using a silken plug near dormant buds on the host tree. So in each one of these little cases, an individual insect. And this is very common, at least in Minnesota, to find them in clusters of 20 to 30 insects around a single bud. As the temperatures warm up in spring, these insects detach their cases and begin feeding in foliage again. So this is the head capsule right here. You can see that dark coloration. This insect has attached its case using silk and then is mined out in this direction and is probably going to start mining in this direction in this picture. So they don't actually, they sort of feed as needle miners in a way, but their whole body remains external to the needle. So kind of an interesting, unique life history here for larch casebearer. When you get thousands of these things on the landscape, feeding on host trees, they can render stands with this burnt appearance. Hopefully that comes through on the slide. And when you zoom out even further, they can create these pretty widespread, large-scale disturbance defoliation events. So this picture was taken by Mark Roberts during an aerial detection survey. I actually think this picture is up in northern Michigan, but we see the same thing in Minnesota. And so not only is Mark taking pictures from this plane, but there's also people sitting with a tablet that are going to document the spatial occurrence of these defoliation events. And so that's what I've mapped out here for you. So each one of these black polygons on within the state of Minnesota is where we detected defoliation between 2000 and 2016. So if you were to look at that through time, we have hectares of defoliation here by larch casebearer on the y-axis as a function of year. You'll notice that I only have defoliation presented here from 2000 onwards. Now, we've had larch casebearer in the state of Minnesota since the 1950s and even earlier, and aerial surveys have been going on for decades, but it just never seemed to reach levels that were detectable during an aerial survey. And the reasons for that were the importation of two parasitic hymenopterans, so these two parasitoid species. And so these parasitoids were collected from larch casebearer's native range in Europe. They were brought over, released. They seemed to reach high enough densities to reduce the densities of larch casebearer below a level that they'd be damaging. And so this is often used, larch casebearer is a textbook example of when importation or classical biological control goes really well. But for some reason, this appeared to I won't say stop working, but as being efficacious as it was in the past, right around 2000, when we've detected now these big outbreaks each year. And so this is about where I came to the project, and I got interested in trying to understand why we might be having this resurgence. And as one might do in this day and age, I started to think about how climate change might be driving this outbreak. So one approach that we used was looking at this cumulative defoliation in Minnesota, and trying to build a predictive model, predicting the presence, absence of where large casebearer defoliation might occur, and what factors govern that. So to do that, if we were to zoom in, and you can imagine this is a disturbance polygon that was mapped during an aerial survey, using a GIS, we overlaid a raster on top of it. So each cell, these were 4 by 4 kilometer cells, each cell that contained defoliation, we assigned it a 1, and each cell that didn't contain a defoliation, we assigned it a 0. So the ones here would be indicated by those gray squares, and the white would indicate a 0 or an absence of large casebearer. And we did this for each year of the data from 2000 to 2016. And so we had a time series at 4 by 4 kilometer resolution across the entire state of Minnesota. We clipped that to where the host tree actually occurs. And then we took to building a model of the presence or absence of casebearer defoliation. So what that looked like is we first fit some temporal terms, temporal lag terms. So essentially this was just to account for the occurrence of large casebearer in a given cell in the previous year or 2 years prior. We also did it to account for a spatial neighborhood terms to account for casebearers in the surrounding 4 cells and in the surrounding 8 cells. So this is essentially just trying to capture a bit of the insect's biology, that places that have defoliation events are typically have areas next to them that have defoliation events, or were likely defoliated in previous years. And this also has a statistical purpose in that it can help capture and soak up some of the spatiotemporal autocorrelation that are in these large landscape scale data. So once we felt like we had accounted for that, those spatial and temporal signals, we took to fitting bioclimatic predictors. And we fit a bunch of these. This is kind of a summary here on the bottom of the slide: minimum temperatures, degree day accumulation. If you're not comfortable with degree day accumulation, you just can think of that as the amount of heat that large casebearers experienced during the growing season. And then we looked at some spring precipitation. So we fit these predictors, and then we developed a best fitting model. And perhaps not surprisingly, we found that having casebearers in a cell in the previous year and 2 years prior is a very strong predictor of casebearer defoliation. So they tend to hang out in the same places on the landscape. And the same could be said for the spatial contagion of this outbreak. So if a cell is surrounded by 1, 2, 3, or up to 4 defoliated cells, it's more likely to be defoliated itself. When we look at our bioclimatic predictors, we found that warmer minimum temperatures in September, October, so sort of less harsh autumn temperatures, tend to be beneficial for casebearers. And we also have found that degree day accumulation tended to be really beneficial for casebearer outbreaks. And so we did a bunch of work on this aspect right here in the laboratory in the field, and I'll kind of just give you a brief snapshot of what the evidence we found and what we think is going on here. So if, as I mentioned, eastern larch is a deciduous conifer. So if you're a large casebearer feeding on this branch as a needle miner, and you're developing in a place and with a host with which you did not evolve. And so you're tasked with developing from the needle mining stage into this autumn case-bearing stage and reaching the overwintering stage before you get dropped into the understory because you're attached to a needle that's being dropped. And so if that happens, you're tasked with not only getting back up to the tree when you may have fallen in standing water, but you're also then feeding on a rapidly disappearing resource. And we did observe some insects in the field that were attached to detached needles that still appeared to be active. So this appeared to happen not only in the field, but we found a signature for it at the landscape scale. We also went back in time, so I want to be clear, this wasn't data that we observed from the 1960s up to the present. This was a model based on casebearer development that we went back and got historical climate data. And what we did is here on the y-axis, you can think of this as the proportion of the populations per year that got enough degree day accumulation to reach the overwintering stage. And there is quite a bit of variability in the signal, but in general, we found that through time, due to warming, a higher and higher proportion of the population appears to be reaching that overwintering stage successfully. And then we found a positive correlation with May precipitation. And we did some work in the laboratory to try and figure out why we might have found this signal, but we haven't been able to find a mechanism for it. We looked at casebearer performance on drought stressed trees and waterlogged trees, and they seem to do pretty well regardless of how stressed out their host is in terms of the precipitation or the water stress that the host is experiencing. So potentially another fruitful avenue of research that we weren't able to quite figure out everything that was going on. So I've been talking about a lot what was going on in Minnesota, and I'm going to jump over now to talking about what's been going on in Western larch. So in Western larch, this is the same graph that I put up of defoliation of Eastern larch, and now I'm going to overlay the map defoliation in Western larch forest. And so you'll notice that the defoliation, as a few years earlier, in 1997, after years of being at endemic levels, these populations appeared to reach sufficient densities that they were detected via aerial surveys. And now the same story, and I'll get into this in a lot more detail, the same story is true out west, that there was a biological control program that appeared to be very successful. And I want to talk a little bit more about the parasitoids before I continue on with kind of those drivers of defoliation. And kind of what we think is going on there. And so the first parasitoid is Agathis Fumila, and this is kind of the star of the show in Western larch forest. It's a braconid, and it attacks the needle mining stage. So here you can see one of those little tiny upside down jello mold eggs, this discoloration here of an early instar larch casebearer, and then the insect is inserting its ovipositor and parasitizing that host. And as a reminder, these life stages are typically on the landscape in summer, the host life stages from summer to autumn. Now, Chrysocharis attacks the case-bearing stages. And it's a different family in the family Eulophidae, but it needs to find hosts that are available in the case-bearing stage in spring, and also in the case-bearing stage in autumn. So there's a large period of the summer where there aren't hosts available for Chrysocharis. And so as I mentioned, the biological control program in Western larch was deemed successful, and it was perhaps one of the most well-studied biological control systems, at least that I'm aware of. And all that work was done by a gentleman named Roger Ryan, who was a research entomologist with the Forest Service in Corvallis, Oregon. And what Roger did was, this is a map here, you can see in the top left corner, Washington and Oregon, in sort of the northeastern portion of Washington and northeastern portion of Oregon and southeastern portion of Washington. So Roger tracked the densities of larch casebearer across these 13 sites for approximately 25 years, while also tracking the parasitism rates of both Agathis Fumila and Chrysocharis laricinelli after they were introduced. And in general, this is what Roger concluded. So you have some measure of moth abundance here on the y-axis. On the other y-axis, you have parasitism rates as a function of time. And so as the moth density was really high, it started to decrease, though, concomitantly with the parasitism rates increasing from Agathis Fumila. And as I mentioned, Agathis Fumila has kind of been the star of the show in the biological control. So this was deemed a success, and a lot of strong evidence for it. And so I was reading this paper, and in the last paragraph of it, Roger Ryan says this: "The limits of my endurance for field work have been reached, and I recognize my deficiencies in theoretical modeling. Should anyone wish to pursue either aspect further, the data will be provided gladly upon request." And the first time I read it, I kind of just thought that's a really cool way to sort of, this is the last paper that Roger Ryan's published, and I thought what a wonderful way to sort of have to drop the microphone on your career. and also a very generous offer to say, I've collected this wonderful data. If anyone, wants to take a further look, be my guest. So I just kind of filed that away, during my doctoral work, and then came across this paper from Miller-Pierce et al., where they are looking at parasitoids in Western larch, kind of trying to understand why all of a sudden there's a bit of a resurgence of larch casebearer out there. And looking at the acknowledgements, I saw that they thanked Roger Ryan. And I thought, oh, I guess he's probably still active. So I reached out to Malia, who then put me in touch with Dave Shaw, who then put me in touch with Roger Ryan, who was super gracious and true to his word in his publication, shared that entire time series of data on large casebearer densities and parasitism rates. So what that means is that we had across these 13 sites this wonderful time series of data. And so what we did is we, made some updated maps. So this is just a zoomed in snapshot here of Roger Ryan's data, or Roger Ryan's field sites. And I've defined what I'll just call as the study area here in that orange polygon. And it'll be clear in a moment why, excuse me, why we define that study area. And that was because we wanted to really look at the defoliation as detected by aerial surveys within the vicinity of the sites that Roger Ryan studied. So here's the graph of the hectares of defoliation log transformed as a function of time. So that orange line is defoliation within that orange polygon depicted on the map, and the black line is region-wide, so defoliation detected across the entirety of Oregon and Washington as attributed to larch casebearer. And so the first thing that I want you to notice on this graph is that after these wasps were introduced, which is indicated by the black wasp on the graph and the gray dotted line. So after those wasps were introduced and established, the populations collapsed in those study areas. There were a few episodes of outbreaks outside of this area within Oregon and Washington, but largely the problem went away. And then I guess right around, so that paper I was talking about with Roger Ryan was published in 1997. So right around the time Roger stopped working on it, all of a sudden we had this resurgent outbreak. So I don't know if that was coincidence or not, or what Roger was doing that was keeping them at bay. But for whatever reason, just like we had in Minnesota, we started to see problems with large casebearer again. So this is the time series that Roger collected, spans from about 1972 to 1975. And so what we wanted to do was look at that historical data and link it with some climate data, and then look at how not only these different hosts and the host in these two parasitoids are interacting, but also how climate is influencing their population dynamics. And so to do that, we defined growth rates for each of them. And what this was a ratio of their population densities in one year divided by their population densities in the previous year, log transformed, which I'll just refer to as growth rates. And so then what we did, we took a two-step approach to modeling. So for the first step, we modeled growth rates as a function of the densities of large casebearer. So looking at interest-specific competition and how that influences growth rates. But then we also looked at the influence of parasitism rates from Agathis Fumila and parasitism rates from Chrysocharis larricinelli. So to use large casebearer as an example, again, we did this for all insects, and I'll present that data, but for large casebearer, we found that intraspecific densities and parasitism were the strongest predictors. So then once we developed a model just looking at the parasitism rates and or abundances of these insects, we developed a reduced model, and then we took to fitting some of these bioclimatic predictors and looking at how climate influences the population dynamics of each one of these species. And I'm going to walk you through each one of those models. But just quickly, the predictors that we were curious about were the same ones that we found were predictive of defoliation out east. So we wanted to specifically focus on these four, but then we also did another analysis looking at some general bioclimatic predictors that I won't go into in great detail, but this is just to say that we looked at a lot of different things, and I'm just going to present the highlights here for you. So for larch casebearer, we found that its population growth was negatively correlated with intraspecific densities. So this means that a lot of larch casebearers in one year meant that its growth into the next year was diminished. And we found the same with Agathis Fumila parasitism rates. So this is very much in line with what Roger Ryan found. You get higher parasitism rates from Agathis Fumila. It has a negative influence on the population growth of larch casebearer. And I'm not going to go into, you'll see this pop up in each one of the models that I'm going to present. I'm not going to go into great deal. It stands for inverse distance weighted densities. So you can just kind of think of that as a variable to account for, again, the spatiotemporal autocorrelation in our response variable here, which is growth rates. And so then we found this signature for warmer October minimum temperatures appeared to lead to faster growth rates, which matched up pretty well with what we found when looking at defoliation on eastern larch. One thing that you might notice is that our strongest bioclimatic predictor on looking at defoliation of eastern larch was degree days, and that seems to be was missing from our final model here looking at growth rates in western larch. And so and Brian Alcama still has a student kind of working on some of these aspects named Spencer Stout, but in general we found that these two populations of casebearer on western larch and casebearer on eastern larch actually have pretty different thermobiologies. So not only are the western larch casebearers more sensitive to cold temperatures, but during certain parts of their life stage, they also develop a lot quicker, which I think might point to the reason that these degree days might not be as important for these western larch casebearers, or at least casebearers residing on western larch to complete their development by the end of autumn, as I talked about in detail for those eastern larch, casebearers residing on eastern larch. So looking at the population growth of Agathis Fumila, we found, and this was a nice thing to find, that high densities of the host of larch casebearer means that their population growth for the Agathis Fumila is accelerated. So that suggests that they should be able to mount some response to increasing host densities, which is something you're always happy to see when you're thinking about importation biological control. And then we found, again, another signature here for intra-specific competition. So if there are a lot of wasps parasitizing the host, they're probably there's not enough hosts to go around for all of them, and it has negative impacts on their growth rates. And then we found this signature for maximum annual temperature. So really, really warm heat events during the summer appear to negatively impact the growth rate for this parasitoid. And in looking at the climatic trends of this predictor, and perhaps not very surprising, maximum annual temperature is getting warmer and warmer as time progresses due to climate change. So potentially a reason why this wasp might not be performing as well as it did a couple decades ago when Roger Ryan was studying these organisms. And so when we look at the population growth of Chrysocharis laricinelli, we find a very similar model to that of Agathis. So a positive response to host densities, a negative response to intraspecific densities. But interestingly, we found this positive correlation between parasitism from Agathis Fumila and population growth of Chrysocharis laricinelli. So you might think, well, these two wasps are competing for the same host. And that might have some negative influence on their population growth rates. But we actually saw the opposite and found that there appears to be some quote unquote cooperation going on between these two wasps. And I'll go into that in a bit more detail in a second, but I also wanted to highlight that again, similar to Agathis, we had maximum annual temperatures seem to really be negatively influencing the growth rate of this wasp. So to talk a bit about that cooperation that we saw between these two wasps, I think it kind of boils down to host parasitoid synchrony. So just going to diagram this out here. You have start of the casebearer growing season on this dot here. They develop through the growing season and need to reach that overwintering stage by the next autumn. So for unparasitized large casebearers, you'd expect it to look pretty simple. But when you're thinking about Chrysocharis females that are foraging for suitable hosts, those suitable hosts in spring are really only available for the first few weeks. And Chrysocharis laricinelli tends to emerge later on in the season. So there's really just a tiny window of time here where suitable hosts are available for foraging females to attack. And the reference here I put up along the bottom has explained this mechanism in detail, but I think that our models might have picked it up. And that's that when you have a case bearer that has been parasitized by Agathis Fumila, so that would be the previous year in the needle mining stage, which would then reach the overwintering stage, and these wasps are going to overwinter inside large case bearer. But those overwintering wasps that are parasitized by Agathis actually emerge several weeks later than unparasitized hosts. And so what this means is that that suitable life stage for Chrysocharis laricinelli tends to have a lot more overlap with their emergence window. And so that they actually have a greater chance to attack hosts and build their populations. And so I think that this positive correlation here picked up on a mechanism that was described by Quaidnow in the 1970s, actually in eastern larch forests. So to kind of put all of this resurgence by larch case bearer together, in a general sense, we had case bearer impacts that seem to be increasing with temperature, which I put here in quotation marks for a reason. Based on our analysis of the Western larch data and looking at population growth rates, we found that October minimum temperatures tended to benefit case bearers. We also found that growing degree days tended to benefit case bearers. But we found that maximum annual temperatures tended to inhibit the population growth of one of the parasitoids. And same story for the other parasitoids. So both parasitoids seem to be somewhat limited by really, really warm summer temperatures. On top of that, parasitism from Agathis pumila tends to benefit Chrysocharis laricinelli. So negative impacts on Agathis pumila that decrease its parasitism rates actually end up having this indirect effect to further benefit large case bearer by decreasing the efficacy of Chrysocharis laricinelli. So I think that I kind of at this point got, this has kind of helped me develop a deep appreciation for how complex these different responses can be by these different interacting organisms to climate change. And what we see here, the evidence suggests that we actually have disparate responses between these two trophic levels. So the herbivore seems to be benefiting from it independent of the parasitoid's performance. But then you also have the parasitoids that seem to be negatively influenced. And so, so we describe a bit of this work and some of the things you can do to contend with larch case bearer in an upcoming fiddle. There is a historical fiddle right now that's available on larch case bearer and western larch, but in collaboration with Brian Alkema and Laurel Havoc, we put together kind of a description of the insect's biology in both eastern and western larch forests and some of the things you can do to manage it. Which unfortunately there's not a lot in terms of silviculture that one can do, but eastern larch and both western larch are planted as landscape trees from time to time, and there are certain pesticides and things you can use to help control them in those settings or for high value trees. So that should be coming out sometime in the next few months, I hope. So if you're interested about some of the case bearer work or more about its biology, I would point you to that. So I'm gonna rapidly shift gears here, and in the spirit of EABU, talk a bit about emerald ash borer and some of the things that I think about in terms of this insect's ongoing invasion and its relation to climate change. So I moved to Mississippi in August, and you'll notice that this map, which here every gray area indicates a state or a province, or a country in the case of Mexico that doesn't have emerald ash borer. Any state that's colored has at least one county that is infested in it. White counties indicate state quarantines, and those red dots indicate where emerald ash borer is. So whatever we're doing here in Mississippi, maybe it's here and we don't know it yet, but it seems to be keeping it at bay somehow. I'm sure it's just a matter of time before it shows up. But certainly across this region, we do have, at similar latitudes, similar climates, we do have emerald ash borer populations. And this is a paper here that kind of flew under my radar until I started to work with Song Lin, my postdoc advisor, looking at essentially the divergence of areas that are suitable for emerald ash borer versus areas that are suitable for its host genus, Fraxinus. And what you'll see here is, this is the EAB infested region depicted here in red. This is the combined ash range in green. And this is a species distribution model indicating the climatic suitability for emerald ash borer. And it does indeed indicate that a lot of those southeastern states, including Mississippi, are suitable for population persistence. So, and this of course shows up in the county level invasion data that I just presented, because of course emerald ash borer is now in northern Louisiana, it's in Alabama, and Georgia, and other southeastern states. But what Song Lin and Liang did was they ran some climatic models and looked at the distribution of, or the potential distribution of emerald ash borer with climate change. And there are a few different simulations depicted here. But even on a short time scale, we show that the suitable areas appear to be receding in the southeast. And so it makes me wonder if in the next 10 to 20 years, if maybe, I don't want to say that emerald ash borer won't be able to persist here, but potentially maybe some of its impacts down here will be diminished compared to what I'm used to seeing and what many of you are at more northern latitudes and across ash forests of the Midwest. So I kind of wanted to present this because one, I gave you a doom and gloom kind of story with larch casebearer and this insect that we had a handle on that then went through this widespread resurgence. Often it's doom and gloom with emerald ash borer, but here it kind of seems like, hey, there's a chance that there could be a climatic refuge for ash along the southern margins of its range as the climate becomes unsuitable for emerald ash borer populations. So that's something that I hope to get into in the next few years now that I'm working down here in the southeast. And with that, I just want to thank you for tuning in. I'll come back to this slide and leave my contact information up. If you have any questions or comments for me, I'd love to hear from you. Of course, I'll do the Q&A session after this, but also at the end, here's a list of references that if you want any information about any of the things I talked about, hopefully this will serve as a jumping off point for you. So with that, thanks again, and I'll do my best to answer any questions you have.
All right, thank you so much, Sam. We have a couple of questions here. This came in at around 11:20. If my understanding is right, your results show that higher rainfall leads to more defoliation. But there are other studies showing different results, that drought or less rainfall lead to more tree mortality or defoliation. How do you understand this?
Yeah, so in the context of larch casebearer, I think that there's just not a huge investment by larch in its not a huge investment by larch to produce a lot of defensive chemicals in its foliage, that because it's a deciduous conifer, it might invest a bit more in growing, because it does have to grow a whole new set of foliage each year. And so I think that there's just not a lot of pressure, at least through the, as precipitation influences on the host, that then scales up to impact the insect. I don't really know how to reconcile why we found that positive signature, but I wasn't terribly surprised that we didn't find a drought stress increases susceptibility to larch casebearer. Now, to point to a different part of that question, we did a different study on eastern larch beetle, which I didn't talk about today, but larch casebearer is viewed as a predisposing agent to that native bark beetle in Minnesota. And we did find exactly what the person asking the question suggested, and that's the areas that are really drought stressed tend to be more susceptible to outbreaks from that bark beetle. But that's going to be more mediated by the not only attractiveness of the tree, but the defensive chemistry of the phloem versus the foliage where larch casebearer is feeding. So I guess to put it succinctly, I don't really know why we found that precipitation signature for larch casebearer, but certainly in other studies that we've done, they line up with what the questioner was alluding to in terms of more drought, more problems with an insect.
Okay, what happens when a LCB is parasitized by both wasps?
Yeah, so great question. And in the case of Agathis pumila, when it is, so there's no chance for Agathis pumila to parasitize a host that is parasitized by Chrysocharis, because Chrysocharis always attacks the case-bearing stage, and Agathis always attacks the needle mining stage, which is inaccessible to Chrysocharis. But when the opposite happens, Agathis pumila always loses out. So if there's a spring activated casebearer that's parasitized by Agathis and Chrysocharis comes along, it will, it can still, and it doesn't discriminate between parasitized and unparasitized hosts as far as we know. So it'll parasitize that Agathis attacked host and it kills the Agathis. 100% of the time, there's no circumstances where Agathis is able to outcompete the Chrysocharis larvae within the host, and then Chrysocharis is still able to develop despite that previous parasitism.
All right. Will the increased temperatures prove unsuitable for the ash tree itself in the south?
Yeah, that's another great question, and I don't know. I know that Song Lin, who is an author on that paper I talked about with EAB, Climatic Suitability, has another paper looking at these for several North American, I think like 87 or something, different North American tree species, showing that there is already some migration in terms of the seedlings and the mature trees over the last 30 years of those, over the last 30 years. And I can't remember exactly what each fraxinus species and what their sort of broad scale migration patterns look like, but that's certainly a possibility. I do think that if you're to think back to that map, there are definitely southern areas of Mississippi that even with the current climate, ash occurs there, but then those areas weren't suitable for emerald ash borer. So of course it's hard to say, but it does seem like there might be even a band right now where emerald ash borer might not be able to develop, but ash does occur. Maybe not at super high densities, but no, that's a great question, and just one of those things that I guess remains to be seen.
All right. Do large case bearers affect all deciduous conifers, such as bald cypress?
No, they're, and I apologize for not mentioning that. They do develop on at least every species of larix that has been tested, but they are specific to that genus. So in the United States, their hosts are western larch, Larix occidentalis, eastern larch, and then alpine or subalpine larch. But as far as we know, they haven't been able to reach high enough altitudes or high enough elevations that those alpine larch occur at. Those alpine larch occur in the same areas as western larch, just at higher elevation. So it's just those three host species that are available for large casebearer in the United States.
All right. Cliff Stadoff has one comment. It says, "Don't bet the farm on Mississippi not having EAB." He also says, any thoughts on physiological changes in larch foliage that could affect casebearer fitness?
Any changes in larch foliage that can affect casebearer fitness? Yeah, so I briefly alluded to looking at how trying to tease apart how precipitation influences larch casebearer performance. And we did that through exposing different trees to different watering treatments and then looking at their defense chemistry and trying to link that to casebearer performance. And we didn't find anything. But there's lots of different aspects of and metrics of host physiology that we didn't look at that could certainly be important. So we don't have evidence for it yet, but but that doesn't mean that there's not still something at play there. And the other thing that we talk about this in the paper that we have looking at these host parasitoid population dynamics, that these models by and large are correlative, and so some of these warming signatures that we're finding could definitely be a heat stress to the host tree that is doing something physiologically that then permits the outbreaks of large casebearer. So there need to be some more targeted controlled studies to pin down the exact mechanisms. But the short answer is no. We're not aware of any changes in the physiology that influence casebearer performance at this point.
Okay. There's a question here that I can probably answer. Are there any predatory insects that prey on emerald ash borer? If you go to the emeraldashborer.info website and then type in the search bar biocontrol, there is an entire web page and also accompanying information and documents that will tell you what predatory insects are going after emerald ash borer and how well that's going. So that can kind of be very helpful for a lot of people to be able to look at that. Brian Akama says, great talk, Sam. You described how it is a race for feeding case bearers to attach to the twigs before the needles are shed from the trees each autumn. Moving forward, do you have any ideas how climate change might affect timing of needle abscission?
I kind of have my guesses, and these are based on like more so warm fuzzy feelings than actual data. So take it with a grain of salt. But I think as it gets warmer and warmer into fall, or if it stays warmer and warmer in fall, I think just what I saw in the field, it seems like the trees do hold on to their foliage a bit longer in the warmer autumns. I don't know sort of two things. One, how much a frost event accelerates that process of dropping the needles. And I also don't know how responsive larch is to photoperiod in terms of its dormancy, which is, of course, going to be fixed by latitude. So that could be something that prevents larch from further adapting to or changing its seasonality in response to climate change. I will say that to the best of my knowledge, and that I don't think larch is really sensitive in terms of its bud break to photoperiod. It's mostly just if it gets warm enough and has sufficient chilling, it can break buds. So, but in terms of the fall, we don't really know about those responses to those different cues.
Okay. What do your models say about the threat of casebearer to eastern larch across its range in the future?
Yeah, so in a broad sense, we haven't done any formal forecasting to show, where we expect outbreaks to occur and that kind of thing. But there were some outbreaks documented in Ontario starting in the 2000s that I'm aware of. And certainly larch casebearer does occur at higher latitudes than Minnesota. So if I had to bet, and based on what our models show, that despite that there are densities at higher latitudes now, that those will likely be able to reach higher and higher and damaging levels that then you might start to pick up during an aerial survey or something as the climate continues to warm.
Okay. Do you think other natural enemies, such as fungal pathogens, etc., would be more resistant to climate change effects?
Oh, geez. Yeah, that's a really good question. I'm a very poor fungal fungal biologist or plant pathologist, so I don't, I'm going to sort of withhold comment, but I think that's a great question.
Okay. So, eastern larch can be planted in urban areas in the Northeast. Can individuals in urban areas be susceptible to LCB?
Yeah, so right outside the forestry building on the St. Paul campus was the first time I found larch casebearer, and it was in really high densities. So at least and that's anecdotal, but yeah, I've seen it on urban. I've seen it on urban sites, and I know I've heard stories, at least on Western larch, of it getting to high enough levels that people have, asked their urban foresters what they can do to treat for it and stuff. So I guess so the short answer is, yeah, I think it definitely can. It just also kind of depends on how much other eastern larch is around, because if it's one larch tree in the middle of a city that's planted with tons of other tree species, there might just not be enough of larch casebearers to establish a high enough population there, or establish one at all. So, but it certainly has the propensity to.
Okay. Do you have any suggestions to understand the interactions between drought and insects on tree mortality, i.e. insects and drought just happen simultaneously, or more insects attack drought-stressed trees, or case by case?
Could you repeat that question for me, Robin?
Sure. Sure. Do you have any suggestions to understand the interactions between drought and insects on tree mortality? And then it says, insects and drought just happen simultaneously, or more insects attack drought-stressed trees, or is this a case-by-case type of interaction?
I would say case-by-case interaction, and I feel like people that work a lot more on tree physiology and what the actual final cause of death is probably have stronger and certainly much more informed opinions on that than I do. In a general sense, at least when you think about in forests, a primary native disturbance agent being bark beetles, I tend to think of those as not being something that that tend to show up on a lot of in areas that are drought stressed. And so I'm not going to paint with a broad brush and say that that's always the case, but certainly that's a well understood mechanism that drought will often precede a bark beetle outbreak. But in terms of defoliators, I think there's my hunch is that there's a lot less work that's done to kind of understand how drought then leads to changes in the insect performance that then lead to an outbreak. Yeah.
All right. We have one more comment from Julie Craves. This has been one of the clearest, most elegant presentations of this type of data/study I've seen. Thank you.
Oh, well, thank you very much for the kind words.
All right, at this time, I'm not seeing any more questions. Folks, you have Sam's contact information right here on the slide, and you will also receive it tomorrow when you receive the email that will have the survey and the information and how to get CEUs for watching this live webinar. Sam, once again, thank you so much. This has been very interesting for a lot of us that haven't even had anything to do with this, and I'm gone from emerald ash borer to doing a lot of more things now. So, and also thank you all for participating. I'm going to end the meeting now, and I hope to see all of you again on the next EAB University session.